So today we will discuss about Southern Blotting. So let's begin with the topic. What is blotting? Blotting are the technique for transferring DNA, RNA and proteins from gel to membrane. So there are three types of blotting. Southern blotting for detection of DNA, northern blotting for detection of RNA, western blotting for detection of proteins. For separation of DNA and RNA, we use agarose gel electrophoresis. For the separation of protein, we use SDS stage. So let's discuss about Southern blotting. What is Southern blotting? Southern blotting is a technique used to detect a specific DNA sequence in blood or tissue sample. So, steps of Southern blotting are DNA digestion, gel electrophoresis, blotting, hybridization in washing, and detection. So, in first step, we digest DNA with the help of restriction enzyme which help to break DNA into fragments. So just look at this GIF in which restriction enzyme comes and attach with DNA, then DNA break into fragments. So in step 2, DNA molecules can be separated according to their molecular weight, and in this step DNA converted into single strand, because in hybridization we need single strand for this step. We use mild alkali treatment, that is NaOH. In step 3, we transfer DNA fragments from gel to membrane. In this step, we use nitrocellulose membrane because it is better for binding purpose. So let's give a look on this GIF in which we have tray in which we pour alkali buffer. It will renature the DNA. Then we took a sponge, then agarose gel, then nitrocellulose membrane. Lastly, we took towel. Then we took any weight like book and press that weight into downward direction. Then capillary action will produce. Then alkaline buffer solution going towards upward direction. Note, everything is on same size whatever we use on this step. In step 4, we incubate nitrocellulose membrane, then many copies of label probes in which all genes are present will aided. This will bind the targeted DNA and form that single strand. We broke in second step into double strand. So in step 5, we detect the bound label probe by using autoradiography in which fluorescence will appear. So this is the GIF of step 5 in which if we detect label probe, then fluorescent will appear and color will change like shown in this GIF. So, what is the application of Southern blotting? The detection of a specific DNA sequence in a mixture of DNA fragments or total cell DNA. So, this is the overview. Step 1, we digest DNA. Step 2, we use gel electrophoresis. Step 3, we transfer DNA from gel to membrane. Step 4, we hybridize DNA. Step 5, we detect DNA probe. Thank you so much for your attention.